All right, well, I just wanted to start by saying thanks for having me on. It's a real honor to be able to present to you all today. I consider myself to be a 11 lab enthusiast, and I think anybody who's met me over the course of the last 5 years, who's let me talk for long enough could attest to that. So with that said, I'll get into it. So my name is Jackson Kubal, and I'm a graduate student at Michigan State, where I work with both the Adami lab and the Bhattacharya lab. Though today I'll just be telling you about my research in the Adami lab. So my project, well, I guess let me give you a little bit of background. So in our lab, we have developed a software called ID-Seek that leverages information theory to solve classification problems. The goal of my project in particular is to use software in conjunction with pure information theory to make predictions about whether cancer cells will be sensitive or resistant to drugs based on their transcriptomic profile. Though, of course, in order for this project to be worthwhile, we want to thoroughly demonstrate that our software makes predictions better than state-of-the-art machine learning methods. This project currently utilizes data from cells in a dish. However, we hope to eventually apply ID-Seek to patient sample data in a clinical setting, thus enhancing personalized cancer therapy and potentially drug discovery. Before we get into my project, though, I just want to cover some background on information theory. I'll start by introducing two commonly misunderstood concepts in information theory, namely information and entropy, and then I'll get into how we are currently applying it in our lab, and then discuss some of our findings and where we plan on going from here. One sec. All right. Okay, so to start, we got to go back to 1948. Imagine you're driving your Cadillac and Frank Sinatra is playing on the radio, but you can hardly tell. Is it Frank Sinatra? Is it Bing Crosby? We don't know. Why does it sound so bad? That's because the channels for radio communication are inherently noisy. They're inefficient, and in addition to that, they're insecure. And circa 1948, there wasn't really any unified approach to solve problems in communication. Meaning if you wanted to fix that radio signal, your only chance of fixing it was really fixing your receiver manually. So born out of this desperation to hear Frank Sinatra without any static or have any foreign spies intercept any radio signals, Claude Shannon sets out to develop a mathematical framework capable of quantifying information. And in addition to that, reducing noise and error in transmission, and also optimally encoding information. So in his paper that he publishes in 1948, he puts forth this framework that allows us to understand what an information channel is. So in it, we have the information source, which you can think of as a song being recorded in a studio. The transmitter, which converts the sound waves into an electric current, which then transmit it over a medium or through a medium. In the case of radio, it would be through the atmosphere, where a receiver then performs the inverse operation of the transmitter, namely converting the electric current back into a sequence of sound waves that you then hear out in your car as a song. So of course, anyone who's ever listened to radio can attest to the fact that there is noise that gets introduced in the process of the signal being transmitted. So with this, Shannon puts forth what he claims to be the fundamental problem of communication, namely reproducing at one point either exactly or approximately a message selected at another point. And he states that in order to solve this problem, you need to quantify and localize the information lost during transmission. Excuse me. So with this, we get to our first term, namely information. So what is information? It's commonly misunderstood. And before we get into any of the equations to define information, I'll just kind of describe some attributes of information. So information is that which provides the ability to make a prediction better than chance. It is the opposite of uncertainty, so rather intuitive. Information must be about something else. It describes a relationship between at least two variables. And lastly, information is symmetric. So given the example from the diagram Shannon puts forth, imagine you have your receiver, which can be characterized by a sequence of electric currents. And you have your song, likewise, the sequence of different sound waves with a relative position. So in this case, we would say that the receiver has information about the song, and likewise, the song has information about the receiver. And knowing the state of the receiver reduces the uncertainty of the song. So here we get to uncertainty and why it's important to be able to quantify uncertainty. And so how does, well, I guess to start intuitively, we already have a pretty good understanding of what uncertainty is. So an event, which I'll refer to as a variable X, capital X, with an equal probability distribution between outcomes has maximum uncertainty. So in the case of a coin flip, the probability of the coin being either heads or tails is equal, and hence the uncertainty of the event is maximal. And then accordingly, if you have an event with no probability distribution between outcomes, then there is no uncertainty. So the probability of the penny still being a penny after the flip is 1, and we would say that this event has no uncertainty. And then so from this, Shannon puts forth his h of x, which allows us to quantify uncertainty, wherein h of x is equal to the negative summation of all the log transformed outcome probabilities weighted by their respective probability, wherein n represents the possible states that x, variable x, can assume, and p of x sub I is the probability of that state occurring. So in the case of the event being a coin flip, we would say n is equal to heads, tails, and the probability of heads and tails accordingly is equal to 0.5. So using log base 2, a unit of uncertainty is a binary digit, commonly referred to as a bit. And sure enough, if we plug in these outcome probabilities into a formula, we see that sure enough, when you have a coin flip and the probability outcomes are equal, namely heads or tails, then you have a maximal 1 bit of uncertainty. And likewise, if you do the same thing, except plug in the outcome probability for the penny still being a penny after the flip, then we see that there is no bits and accordingly no uncertainty. So upon putting forth this this equation to quantify uncertainty, Shannon famously goes down the hall at Princeton, I believe, where he bumps into none other than John von Neumann, where he asks von Neumann what he should call this equation, to which von Neumann responds, you should call it entropy for 2 reasons. In the first place, your uncertainty function has been used in statistical mechanics under that name, so it already has a name. And in the second place, and more importantly, no one really knows what entropy really is, so in a debate, you will always have the advantage. So of course, it's worth pointing out here that von Neumann is actually lying in this quote, because there was in fact one person, at least at that time, who understood entropy. And it was none other than von Neumann himself, who actually put forth the P log P formulation of entropy in 1927, about 20 years before Shannon, except he used it for quantifying the uncertainty of disorder in quantum states. I could go into more detail about the similarities between thermodynamic entropy and informational entropy, but I'll save that for another presentation. Just know that there is a stark similarity between the two. So here we get basically just the graph that Shannon actually puts in his initial paper, characterizing the relationship between entropy and probability outcomes, essentially characterizing that when outcome probabilities approach 0.5, or in other words, random chance, you have maximal uncertainty. And then likewise is true as you move away from 0.5. the amount of entropy in the system moves towards 0 and 1. So given this formulation of entropy, the question then is, what is information? Well, put it simply, information is just entropy that is shared between variables. So in the example where we have the receiver, just a sequence of electric states, and the destination, which we can represent as a song, and respectively as X and Y, we would just simply say that the information between X and Y is equal to the entropy that's shared between X and Y. So that those colons denote between. And then we can say the amount of information between X and Y is equal to how much h of X is reduced by knowing Y. So we just write that mathematically as h of X, the entropy between X and Y is equal to the maximal uncertainty of X minus the uncertainty of X given Y. Where h of X given Y is essentially just a slightly modified version of that equation I have at the top of the slide, wherein instead of just plugging in the probabilities of X, we just condition those probabilities on us already knowing Y. So instead of it just being P of X sub I, it's P of X sub I given that we know what Y is. Thus, we could state if h of X given Y is less than h of X by itself, then Y has information about X and vice versa. So now we can apply this to actually understanding information channel and where information resides in the channel. So now imagine you have your receiver state or your receiver states, and you have your relative position on the receiver. So just imagine a sequence of different electric states on the receiver currents, whether or not it's on or off, essentially. And then we can quantify the entropy of that by just plugging in the distribution of states, wherein we see that there's an equal probability distribution between states. So we would say that this receiver state is maximally uncertain. In other words, there is 1 bit of entropy. And then we can do the same thing for the destination for the song. And we plug in those probability distributions, and we see, sure enough, that there is 1 bit of entropy. And now what we could do is essentially apply those formulas we just learned to figure out what is the informational relationship between receiver and destination. So we just plug in those values. So we see that h of X, the entropy between X and Y is just equal to the entropy of X by itself minus the entropy of X given Y. And by just looking at the states of the receiver and the destination in this example, very straightforward, we see that when we know Y, we can predict what X will be with perfect certainty. In other words, when Y is 1, X is 1. When Y is 0, Y is 0. So the probability of our predictions will be 1. Therefore, the uncertainty of X given Y, the pipe denotes given, just a conditional, is equal to 0. So therefore, the shared entropy between X and Y is 1, and the information between X and Y is 1. So all of the entropy between receiver and destination is shared, and therefore we would say that this is a noiseless channel. So now bringing it back, we can see that if given 2 states in a information transmission channel, we can now apply information theory to understand where that information is. If we know X and Y, if we know any 2 states of in the system, we can calculate the conditional entropies, h of X given Y, h of Y given X, because it's symmetrical. And then we can figure out the informational relationship, and we can quantify and localize specifically, regardless of whatever transmission channel we're working with, essentially where that information is and where it's being lost. So given this, Shannon then puts forth his error correcting code, as well as the possibility to encrypt information so as to secure it. And in that, we see where the application of information theory kind of comes from. So that's all great, but now you're probably wondering, well, how does this all apply to biology? To which I would reply, it doesn't just apply to biology, but rather it is biology. So by definition, life is that which self-replicates in formation with its environment. If you can't accurately transmit and store information about your environment, then you're not going to be able to survive for long. So in order to do that, organisms, not just organisms, but really biological units, because it scales all the way down to a suborganismal level, have had to transmit information internally, externally, and temporally for the last 3.8 billion years. And if you aren't able to do that, then, yeah, like I said, you're not going to stick around for long. In fact, what makes an organism fit is, by definition, its ability to store information about its environment. So here I included this in here as a canonical example of information transmission in biology. And if you have ever taken a molecular biology class, you've probably seen this. And it's the lac operon, which is just a gene locus in E. coli bacteria that is either on or off, dependent upon what nutrients are available to the cell. So if you just take basically my receiver and destination example in radio communication from earlier and plug in glucose availability and lactose availability, and then plug in gene expression levels instead of the song, then you have yourself an information transmission channel, wherein knowing the states of glucose and lactose gives you a high fidelity predictive power about the state of the gene locus. The problem, of course, is that just like in radio, excuse me, these information channels exist in extremely noisy environments. So in the case of E. coli, its primary surrounding environment is the human GI tract. Thus, in order to accurately transmit information in this environment, E. coli essentially needed to figure out how to encode and encrypt information. So really, I think it's safe to say that when Claude Shannon proposed the fundamental problem of communication, what he was really doing was capturing a problem that life has been dealing with for the last 3.8 billion years. And just like Shannon, or rather Shannon, just like life, figured out that in order to deal with these issues, it was necessary to encode and encrypt information. And so what do I mean by life coding information? Well, suppose that you have a small colony of E. coli cells that are hanging around in the human gut, and in the presence of glucose, the quantity and the location of the glucose is captured by this sequence of cells in the colony. We can think of each cell as being either in an on or off state. And recent research, I think at UCSD, I'm sure other people who are doing this as well, has shown that bacterial biofilms are actually capable of storing collective memories in their individual constituent cell membranes. But anyways, the important point here is that in order for the individual cells to know what state they should be in, namely whether or not to grow or to sequester nutrients, it is imperative that accurate information about their environment be transmitted. It's in the cell's best interest to accurately provide information to their neighboring cells, and vice versa. So in order to ensure that information is transmitted accurately, the cells essentially redundantly encode their state space in their gene pathways. So what do I mean by that? Well, instead of having each cell represented as just a 1 or a 0, cells encode, they blow up their state space into the different gene pathways. And now instead of just having one signal being transmitted, you can transmit 5 signals. And then just using a majority vote, you can decode that error in transmission, assuming that there is a 1 in 10 chance in this example of your signal being flipped in the process of transmitting. They can essentially error correct that code by just redundantly encoding it in their gene pathways. And then with a majority vote, decode that signal and recreate a high fidelity state space of the cells. So of course, the price that organisms pay for this is the rate of transmission slows down, but by blowing it up, by blowing up the message into the different gene pathways, the cells can reduce error so from about 1 in 10 states transmitted errantly to approximately 8 errors in 1,000 states. And if they were, in this example, to encode it in 10 different gene pathways instead of just 5, they could reduce error down to approximately 1 error in 10,000 states transmitted. So I said life encodes information, but life also encrypts information. So a classic example of this is in the codons of an amino acid. So of course, this is also an example of encoding information in the sense that the information is redundantly stored in the genetic code as opposed to the information being stored directly in the proteins themselves. But codons themselves, as we know, don't provide all the information necessary to predict protein expression. In order to know if a gene is transcribed, you need to know the epigenetic code on the DNA. And furthermore, there are genes, especially present in viruses, where you actually need to know the physical structure of the RNA molecule in addition to the codons themselves, if you want to accurately predict the resultant protein. So it's worth pointing out here that codons themselves actually have information about the environment that you wouldn't be able to predict from the amino acids themselves. So, and that's because different environmental selective pressures favor usage of different codons. So in different organisms, we have different codons being used, and that's because different selective pressures at different levels are selecting for different codons for different reasons. So this isn't really anything new, though. This is fairly well understood all conceptually. So what we wanted to ask with this project and explore with this project is information encrypted in the transcriptome and in the transcriptome alone? So how do we go about doing that? Well, we just start off with asking the simple question: is a cell going to be sensitive or resistant to a drug? And we determine that based on by essentially binning all the cell lines into 2 groups, sensitive and resistant, wherein we base that off the IC50 score for the respective cell line. And then we bin the gene expression levels for the respective cell lines into on or off. And we did that using just RNA-seq data. So given this formulation, we can then ask the question: how much information is shared, or how much information do the transcriptomes of the cell lines have about the drug response? In other words, how much entropy is shared between the two, or how much entropy resides in that middle sliver of the Venn diagram. So the standard approach for a question like this is, of course, to just do differential expression analysis, wherein you just take your 2 groups of cells, in this case, sensitive and resistant, and then you look at the principally differentially expressed genes. When I just doing my my differential expression analysis, I see that this gene ALF1 or IF1 comes up as the #1 most differentially expressed genes between the 2 groups. And then what we would do typically, or what most labs I'm aware of would do typically, is they would take this list of differentially expressed genes and rank them based on how confident the DE-seq is in this case, and then rank them. So that's what I did here is I basically took all the genes that are differentially expressed between the two groups and then ranked them in terms of confidence of the basically how confident are we that these genes are truly differentially expressed, and then along the y-axis we have the respective p-value ranking for those genes. And then if we wanted to train a neural network to make predictions or a random forest classifier, what we might do is take, say, the top 200 genes or top 100 genes, depending on what you're using, and then try to train a model on those, and then call it a day. We would look at those genes, analyze them, and that seems to be about the standard approach, to me at least. And then with that list of ranked genes, we can go a step further. We can rank them not based just on their individual expression levels, but we can kind of look at trends between groups of genes that are expressed similarly in the different conditions, and we can look at the principal components that explain that variance. And then we can even use that to do a rudimentary classification, which in some cases is more than enough because the transcriptomic signatures of the different cell lines are so distinct that it becomes obvious which cells belong to which groups. But in a problem as convoluted as this, we see that it obviously is not a great classifier. So from that, we get into where information theory and ID-seq shines. So I think the reason as to why the standard approach fails and why utilizing information theory is so advantageous is perfectly characterized by this C.S. Lewis quote, in which he says, "Of three friends, A, B, and C, A should die, then B loses not only A, but A's part in C, while C loses not only A, but A's part in B. In each of my friends, there is something that only some other friend can fully bring out." And I think this quote so perfectly captures how information can be encrypted in the transcriptome. And all you have to do is just change where he says friends to genes. So now we see that essentially in each of my genes, there is something that only some other gene can fully bring out. So what does that mean in terms of information theory and ID-seq? Well, suppose we have a gene X, and we have our class, which will represent with sensitive and resistant, SR, and then the gene expression levels, high and low. So given this, we can look at how much information is shared between the gene, or how much information gene X has about the class. And doing that, first we see that the entropy of the gene is 1. It has an equal probability of being high and low. So we would put that 1 there. And then we would see that class likewise has an equal probability of being sensitive or resistant. So a 1 there. But we see that knowing the gene doesn't give you any predictive power about the class and vice versa. So we would say that the shared entropy between gene X and the class is 0. What information theory allows us to do is explicitly define the information that X has about class in the presence of another gene, gene Y. Oh, sorry, I get ahead of myself. So this is what we refer to as first order information. So in this case, we would say that there's 0 bits of first order information between gene X and the cell. But like I said, what information theory allows us to do is now condition the entropy between X and class on the presence of another gene. So now we can introduce gene Y, and we can see that gene Y also has 1 bit of entropy. It has an equal chance of being low or high. But what we see is that gene X in the presence of gene Y actually, which I represent in that upper sliver of the middle of the diagram, becomes 1 in the presence of gene Y. And what does that mean functionally? That means that given gene X and gene Y together, all of a sudden, we have perfect predictive power about the state of the class. In other words, when gene X and gene Y are both high, excuse me, when they're both high or they're both low, then we know that the class is going to be resistant. So the entropy of the class drops down to 0, and the entropy of gene X and gene Y also drops down to 0 in the presence of the other two. So now we can say that the entropy of class given X and Y is 0, and there is one bit of what we refer to as second order information between gene X and the cell given gene Y. So, oh, sorry about that. So that was obviously an example, but to our knowledge, this is not something that has been thoroughly tested. And so what we wanted to do is say, okay, well, does that actually happen to be the case in this particular case? So looking at these cancer cell lines, do we see any information stored in pairs of genes that isn't inherently stored in individual genes? And so we did a screen of just 100 genes, just a base level screen to see, is there any information stored in X and Y together that isn't stored in either of them by themselves? And that center sliver of the diagram, that -1, that represents encrypted information. So sure enough, we ran our analysis, and if you see that the mode is shifted, in fact, to the left. So there is negative information. In other words, there is encrypted information stored in gene pairs that gives us more predictive power about the class. So that was reassuring. But that's just looking at second order information. So there's no law that says there can't be information stored. So like I said, that's second order information, which in other words is entropy between two variables and the class. But there's no law that says there can't be information stored in even higher order pairings. So if we introduce another gene, now we have third order information about the class. And then why stop there? We can go on to fourth order information, where in which we use 4 genes in combination with each other to predict the class, and so on. So in theory, the data set I'm working with, has 18,775 genes. So in theory, we could run this analysis all the way up to order 18,776 and potentially find information that is encrypted all the way up until that order, wherein you're not going to see that information until you take in the whole entire transcriptome together. But of course, you would need a data set that is probably like bigger than the amount of atoms in the universe to have statistical significance for doing that level of decrypting. But to get to these orders, we have plenty of cell lines. So now what we did is we basically implemented an information theoretic based algorithm that simply iterates through all the genes. And then we use those to identify the genes that contain information that is stored in the higher order pairings, and gives us predictive power about the class. So using that, we identified 8 genes, which we refer to as the jury, that when taken in conjunction with each other, give us maximal functional information about the class. So essentially what this means is you're not going to be able to make any better prediction using any other set of genes in the transcriptome about the class than you would be able to using these 8 genes. So who are these 8 genes, which we refer to as the jury? So jury number 1, THBS1, this angiogenesis inhibitor. Jury number 2, we're not a cancer biology lab, so I don't know exactly the full extent to which these are implicated in cancer biology. But just by doing a quick Google search, I kind of generated some of these descriptions. But juror number 3, and then we get all the way down to our full jury, our full cast of characters that are capable of making a maximally informative prediction about the class. And what's noteworthy about this list is that really only 3 of these jury members, namely THB S1, TSP OAP1, juror 1 and 2, and then juror number 7, HCK, are implicated in drug resistance in cancer cell lines. So that means that these other 5 members could be potentially novel genes that up until now have not been implicated in any cancer drug response pathways at all, simply because they haven't been taken into consideration in conjunction with the presence of other genes in these drug response screens. And then, of course, I have on the right a picture here of the jury deliberating about the fate of the cancer cell line as all the scientists look in the other direction, bamboozled by the cancer cell line's lawyer. So from that, we obviously want to ask, okay, so these 8 genes are highly informative about the class, but where do they stack up in compared to our initial differential expression analysis rankings? Because we would obviously assume that these highly informative genes would be all, highly differentially expressed, and probably the most, if not in the top 8, right? Well, it turns out that that is not the case. So when we take these genes into combination at higher orders, the number 1 ranked gene in the jury is ranked number 200 and is ranked number 400 in the list of differentially expressed genes between these 2 groups. So what does that mean? That means if you were to be doing feature selection to train your neural network or your random forest classifier, and you were to stop at 200, the top 200 genes, you would miss all of the most informative genes capable of reducing the entropy of the class. Of course, you could still make a pretty good classification, but you're not going to be making the best prediction possible. And what's fascinating is that once you get into the end of the jury, we get 1 gene that's ranked at approximately 4,500 in terms of just standard differential expression analysis. And then the final 2 don't come in until about 15,000. So this means that you would have to essentially include the whole transcriptome in your analysis if you wanted to not miss out on these informative genes that, when taken into consideration in the context of the presence of other genes, are in fact highly informative. And why is this the case? Well, it's the case because some genes only contain context-dependent information. Namely, if I were to only look at LAG3 and pCCA by themselves, I would have no my ability to predict the class would be as good as random. However, if I look at LAG3 and pCCA in conjunction with those first 7 genes that we already know are highly informative, then all of a sudden those genes become highly informative themselves. And so what does this mean in terms of actually making a prediction? So here I represent 3 different predictors, which is a random forest classifier, a standard tool used by a lot of different labs, and a neural network, and then we have IDSeq, our software. So this is just our software, IDSeq at order 1, meaning it's only looking at the information contained within the individual members of the jury. And we see here that the neural network outperforms all the other ones at just 1 gene, but then by all the genes, once all the genes are included, then they're essentially equal in terms of prediction accuracy. But with just one gene, it's not surprising that the neural network does so good, because for the neural network, we don't have to bin any of the data. Same thing for the random forest, but for IDSeq, we do bin the data. But now we can take into consideration order 2. And we see it's still not doing much better than order 1, and definitely not better than a random forest or neural network. Then we can go a step beyond, go to order 3, and we see definitely an improvement over order 1 and 2, but not much better than the neural network or the random forest classifier. It isn't until we go to order 4 that we see an about 4-point jump in prediction accuracy. And what's more, if we take it one step further and we go to order 5, we can actually go above 0.8, and we see a 6-point jump in prediction accuracy when we utilize our IDSeq at order 5. So an AUC of 0.8 isn't it's something crazy. However, what is worth pointing out here is that the error in the IC50 scores, that in the Z-scores that we're using to bin the sensitive or resistant classes, has error built into it already. So if we remove a lot of the cell lines that have a Z-score very close to 0, that basically, yeah, if we remove the cell lines that have a Z-score very close to 0, then we can essentially improve the accuracy of this prediction all the way up to a perfect classifier. It's just because there's noise inherently built into the IC50 measurements. But so with IDSeq at order 5, we are able to increase our prediction accuracy by 6 points. So we find this to be rather exciting. So what is it, in summary, that is giving information theory the predictive edge here? Well, it explicitly captures information that is distributed over many variables. Other methods don't do this. So if you're using a neural network, there's a good chance you'll pick up on some of these higher order correlations between variables. However, there's a chance that it misses it, because it's ultimately what you're doing is you're fitting a probability distribution to your data, whereas information theory does not attempt to fit a probability distribution. Information theory calculates probability distributions directly from the data presented. And so, in essence, it's a principal way of not overfitting. So in this example, in this project, what I've used our software and information theory to do is make a prediction about drug response given the transcriptome. And in addition to that, characterize some potentially novel, insightful, biologically relevant pathways. And why is this important? Well, I think that speaks for itself. This is a potential avenue for novel drug design, as well as patient screening, by basically characterizing these relational structures that exist between the different genes. We can actually unveil deeper insight about things like drug response, but it doesn't actually have to stop there. We can look at membrane voltages on cell lines in conjunction with transcriptomics to make predictions about morphology. We can make predictions about whether or not disease will arise based on membrane voltages and morphology, try to predict developmental outcomes. The list really goes on. The point here is that we're really only limited by what sort of measurement devices are available to us. So whatever information is present within a classification problem, we can discern it maximally. And so the reason why this is so advantageous, at least in our lab's view, is that the chances are, and it seems to be apparent that this is the case, that the primary information containing signals are not going to be a point mutation, and they're not going to be a single gene pathway. They're not going to be a single protein, but rather they're going to be a distributed signal that is contained within multiple different transmission channels. And so in order to discern that signal, you're going to need something that can explicitly define those relationships. So in closing, I want to leave you with a quote that you're probably familiar with that I came across when I was listening to one of your talks earlier this week, where in which you say biomedicine and bioengineering boil down to finding out the informational structures of the material of life. And I couldn't agree with this more. And I think that it's more often than not going to be a single variable that is informative, but rather the higher order correlations between variables that unveil the information necessary to devise novel life forms like you guys are doing in your lab, and also to address diseases such as cancer, as well as come up with cures for those diseases. So with that said, here's the resources from where I got the data, transcriptomes, from the link at the top, and then the drug response data from the link at the bottom. So feel free to check that out. And lastly, and most importantly, in addition to Shannon and von Neumann, I would like to thank these three gentlemen, for whom without this project would not have been possible. Chris Adami, who wrote the book on applying information theory to biology, Joy Chassau, who built the foundation of IDSeq, and Vinny Ragusa, who continues to improve IDSeq today and has also been a tremendous help for me in this project. And I'll lastly leave you with this. But that's really the end of my presentation, and I hope you all enjoyed. And please reach out if you have any questions, and let's do some collaborating.
Super. Thank you so much. Questions, comments, ideas, everybody?
I have a quick question. Great presentation, by the way. Maybe I missed this. How did you arrive at the number 8 out of the 8 genes from among the 18,000 odd genes? What is the method that you use?
Yeah, so that comes down to just the statistics of the data set we're working with. So 2 to the power of 9 is 512. And at that point, what we're really doing is just hashing the cell lines. So you can basically uniquely identify every cell line in the data set by going to order 9. But at order 8, we're still, we believe, picking up on biologically relevant data that gives us insight into what differentiates these two classes. Because 2 to the 8 is only 256. Beyond that, we're essentially, you'd be overfitting. It wouldn't be, any of the genes wouldn't be able to reduce our ability to predict. Any more genes are no longer enhancing our ability to predict. They're actually hurting it, because they're no longer capturing differences between resistant and sensitive cell lines. They're capturing differences between individual cell lines.
Oh, okay. But how did you arrive at that particular set of 8 genes? Like, how did you sift them from the lot? That's
so we implemented an information theoretic based algorithm, and unfortunately, I don't think I'm able to disclose that algorithm, because it's yeah, MSU.
all
you
right,
know.
got it. No.
I think it was a mind-blowing talk. Just a quick question. I mean, so estimating an entropy requires you to know what's the probability distribution of the random variable, and how did you estimate it?
I'm sorry, I couldn't really hear you.
Yeah, how did you how did you estimate the probability distributions for the sake of computing entropy?
The probability distributions of the different genes.
Yep.
So gene expression levels are just continuous distributions, right? They could be anything, because they're just based on RNA counts data. So what we did is we binned the genes into 2 different bins, essentially trying to maximize the entropy of the individual genes. but meaning like to split the two groups of genes into two equally populated bins, but namely on or off.
Thank you.
Right, but and to keep in mind, then basically we're doing a multiple sequence alignment, or here a multiple alignment of the cell lines. That to order one is just looking at, well, which of the genes are most expressed in one or the other, and then you go do order two, order three. We basically create position weight matrices. So in other words, we don't actually, these probability distributions just can be read off of this, alignment. So there's no fitting of the probability distributions. They simply, can be calculated directly from the data. And in fact, in a sense, is the slide that you see here is how we are calculating this probability distribution. On the left side, you see this comment on social media where they're saying, oh, what makes machine learning so intractable is because you cannot calculate the partition function. And that's what essentially is being fitted. But it turns out that you can calculate it. And here's the calculation. And in fact, it's calculation to, as an approximation in higher orders. You see the first order and the second order, and you don't see the third, fourth, and fifth order. And so we essentially calculate the partition function using this sequence data. And there is not a single step of fitting in the procedure, which is why, we don't overfit noise, right? So the entire secret really is about the fact that, this statement is simply false. You can, in fact, calculate this probability distribution directly from the data. It's just that nobody knew about it. That's all.
Franz, did you have a question at one point?
Yeah, I was wondering if you can use that IDSeq framework to infer the entire network from the RNA-seq data. Is something you guys have tried?
That's something that's currently underway. So we have networks already. I didn't include them in my slides of taking into consideration basically relationships between genes at order 2 and seeing basically what those genes are. But further work is required to go beyond order 2 correlations, and then also in actually contextualizing them in a biological context and seeing like what does this mean biologically.
Okay.
How important do you think are so if you were to include other kinds of data, how important is it the exact way that you, let's say, bin the data or map it into ways that your formalism can handle? Is that really critical, or is it pretty insensitive to that?
It's definitely sensitive to it, but it's really I believe it would be a case-by-case basis. So it just depends on what exactly the data is. So in this case, we're obviously looking at transcriptomes, so they're all in the same realm of compatibility with each other. Leveraging different types of information, different pools of information together would require further research.
Yeah, but I think we can generally say, and this is work that Vinny has done, that I mean, we've tried a number of different ways of mapping data in different modes, in particular, non-digital data into digital data. And the approach that seems to work best is if you basically say that without any class label, that means for all of your data, just bin it in such a way that each feature has the highest entropy, basically that you have, it's a maximum entropy binning, in the hope that when you then apply the class label, that would maximally reduce the entropy. And that, I don't have really a mathematical proof, even though maybe it's not that difficult. It's literally like maximizing the channel capacity, if you're doing this. So my suspicion is that in most cases, that is going to be a good approach. But then again, we might even imagine that you, case by case, use a different number of variables. So in other words, we're doing binary binning here for transcriptomics, but you could say, why not base 4? In other words, you distinguish not just high and low, but very high, medium, low, very low. And it turns out that some genes actually have quite a bit of variation in the profile, so that you would treat different informational structures with a different number of bins, right? They don't have to all be binary. So you have a hybrid method, but in both cases, you would probably use a maximum entropy, but you would make a feature-specific, thresholding, right? Because it really depends on how broad that probability distribution is that you're trying to bin. It turns out that in transcriptomics, binary is doing pretty well. The reason why we're doing binary is that as you're going beyond binary, you're going to have hard time capturing the higher order information, right? If you go base 4, well, you're not going to be able to look at 8 genes anymore, right? You're probably going to be stuck at 4. And then many of the genes probably don't need a resolution of 4. But we've, like I said, we've tried different methods. I think Vinny has spent easily a couple of months working on what are the optimal methods, and the maximum entropy binning method appeared to be the most promising.
Got it. Okay, good. That's great. That means we can think about some of the other kinds of data sets that we have, some of the physiological data sets. Yeah, that's great.
Yeah.
Okay.
I mean, Vinny even has looked at data where there was patient data like age and what was it? I mean, educational background. I mean, your typical patient data, right? And he was able to use that, bin it appropriately, and then use it together with the microbiome and with with gene expression, transcriptomics and stuff like that. So in other words, it really doesn't matter. If you think your data contains information, just throw it in. Mm-hmm. And you might actually see interactions between microbiome and transcriptome. And mm-hmm. and we saw strong interactions between patient health data in some of these variables that are genetic, and variables that are, this was an Alzheimer's study, like plaque thickness, and tau protein abundance, things like that.
Mm-hmm. Got it. Amazing. Okay, cool, guys. Thank you very much. Let me ponder this, and we'll be in touch. Let me think of a few data sets that we could work on.
All right.
That'd be great.
Yeah, very interesting. Yeah. Thanks. Very cool.
Thanks for listening, guys. I hope everyone enjoyed.
Absolutely. Yeah. Good to meet you, Jackson. Good to see you again, Chris.
Yep.
Good to see you, Mike.
Bye.